Hello everyone, my name is Kwanghyun-jo. Today I'll talk about computational modeling and analysis of cellular signal transduction pathways. Today you will learn what signal transduction system is and how to describe the underlying biochemical reactions in mathematical terms. And then in your lab, you'll try how to do the mathematical modeling. The basic unit of life is a cell, and a cell has developed a signal transduction system to sense external changes or perturbations and transfer the information from the outside of the cell to the inside and to make a decision for cell fate determination. So signal transduction is a process by which a cell transfers one kind of signal or stimulation into another. All these processes are actually very fast, but some of them takes a longer time. Enzyme is one of the very important element in signal transduction pathways. Enzyme, as you know, it's basically a catalyst that help converting one molecules called the substrates into product. But they do not change by themselves during this reaction. Three important features of an enzyme are catalytic power, specificity, and regulation. The mathematical modeling of these molecular reactions mostly based on the law of mass action. The assumption is like this. Molecules are assumed can freely move around, and if they collide with each other, then there is a chance to produce another product. So if we consider two molecules, A and B, then if they collide together, then they can produce another complex molecule, C. We can describe this using this forward reaction. So based on this assumption, we can think the rate of change of the complex molecule is proportional to the concentration of both A and B, and the proportional constant is K. If we consider bidirectional reactions, that is not only the forward reaction but also the reverse reaction, then you can further describe this reaction by adding this reverse reaction terms. So if we solve these equations, then at steady state equilibrium point, we can obtain this equilibrium concentration of C, or for both forward and reverse directions, we will have this one. As you see here, if we plot this curve, then it starts from 0 and converges to the steady state concentrations. We can further extend this idea in general way. So let's think of two reactants. They make a reaction and produce the same type of two products. So for example, if S1 and S2 react together to produce two products, then we can write down its a reaction equation using the mass action law. Then the rate of change of the production rate of complex will be 2V. On the other hand, the rate of change of each substrate molecule S1 or S2 will decrease as much as minus V. We can further generalize this idea. So if we consider these reactions, then the rate of change of S1 will be minus 2V, S2 minus 3V, and P1 will become 2V and P2V. So we can write down the total rate of change of the reactions in this way. So there is an increasing term that is proportional to S1 and S2, and decreasing term proportional to P1 and P2. And there is a index proportional to the number of molecules being used for this production and decrease. We can generalize this idea. So the general rate equation of a mass action law will become the production term minus degradation term. So here Mi and Mj denote the respective molecularities of substrate and product. So this one refers to the production rate of a complex, and this one refers to the reaction equation of reactants. Let's think of more specific biochemical reaction made of enzyme and substrate molecules. Let's think of an enzyme E converts the substrate molecule S into the product P through an intermediate step by forming a complex C. Then the whole scheme of biochemical reaction will become like this. So here enzyme and substrate bind together, and then it forms a complex. After that, the substrate changes into product, and enzyme returns to its original state. If we just think of one of these, for example, substrate, the rate of change of a substrate molecule will increase as much as this backward reaction of K2 and proportional to the concentration of complex. On the other hand, the concentration of S will decrease as much as the production rate of complex C. That is this term. So if we repeat in this way, then we can write down all the mathematical equations for substrate, enzyme, complex, and product. Here, we can assume that the total concentration of substrate and the total concentration of enzyme, they do not change. Then usually the enzymes are saturated with the substrate. That is, the concentration of substrate is usually much larger than that of enzyme. So in this case, we can assume that the complex is at an instantaneous equilibrium point that we call quasi-equilibrium state. In this condition, the rate of change of the complex becomes zero because there is no more change with respect to time. Then we can simplify the previous equation in this way, and then by solving this algebraic equation, we can obtain the concentration of complex becomes like this. So here, capital K sub M denotes the Meares-Manton constant. Because the rate of change of the product is proportional to this complex concentration, and the proportional constant is K3. So if we summarize this again, then we can finally have this Meares-Manton equation that describes the relationship between the rate of change of the product with respect to the concentration of substrate. Here, Vmax indicates the maximum reaction velocity, So you can imagine that this rate of change of the product will saturate to the point Vmax if S goes to infinite, right? And then for S0, it will 0. So the curve will become like this. And if the substrate concentration is Km, then this equation becomes just 1/2 of the Vmax. So that point actually a measure of a speed. Such enzyme substrate reaction is often regulated by inhibitor. There are two types of inhibition. One is competitive inhibition, and the other one is allosteric inhibition. Competitive inhibition means that usually the enzyme is much larger than the substrate molecule. So if we think of one specific substrate enzyme binding site here, if there is a very similar structure that we call inhibitor, if it binds to the same binding site, then it can prohibit the binding of the substrate. So the inhibitor and substrate competes together for the same binding site. This is competitive inhibition, and we call such inhibitor as a competitive inhibitor. On the other hand, the binding of enzyme and substrate can actually modify the three-dimensional structure of enzyme. That is, without binding of inhibitor, normally the substrate and enzyme binds at this site. But if there is an inhibitor binds to the inhibitory binding site, even though it is far from the substrate enzyme binding site, it can cause some conformational change of the enzyme and substrate binding site. So it can inhibit the binding of a substrate to enzyme. So this is remote control, and we call this allosteric inhibition. And such inhibitor is called allosteric inhibitor. We can describe this reaction in terms of the following mathematical model. It's very simple. The reaction scheme is like this. So as you see here, the substrate binds to enzyme without any inhibitor. But if there is an inhibitor, in this case competitive inhibitor, then because there is only one binding site, once enzyme binds to the inhibitor, then the substrate cannot bind to the same site. So if we write down this reaction in terms of mathematical model, then let's just think of this one, complex 1. The rate of change of complex 1 would be proportional to the product of substrate and enzyme concentration multiplication because of this forward reaction. And then it is inversely proportional to the reverse reaction of K2 and K3, like this. So we can finally have the rate of change of C1 in this way. Similarly, we can have all other mathematical equations. And then, again, we assume the same enzyme to substrate quasi-steady state condition. At this point, the rate of change of both complex formation will become 0. Then we can simplify the previous mathematical model, and if we solve the equation with respect to complex 1 and complex 2, then we have this equation. So if we look at the rate of change of the final product, then we have this one. Oh, this is very similar to the previous Meares-Menten equation. The only difference is here. So if this is just 1, then it is exactly the same as Meares-Menten equation. Here the only difference caused by this inhibitor term. So because of this inhibitor, Km is effectively larger. Because Km indicates the response speed. So you can imagine that this reaction will result in the same level of maximum speed. On the other hand, it only changes the response speed and it becomes much slower. So you can see that the competitive inhibition actually results in much slower response, but with the same maximum speed, maximum rate of change. In the next, let's think of allosteric inhibition. It's again very similar to the previous one. The only difference here is, here we have an enzyme. Because it has 2 binding sites, normally it binds to the substrate. Without inhibitor binding, it produces product, and the enzyme returns to the original state. But if there is an allosteric inhibitor binds to some remote site, then this enzyme and inhibitor binding actually prohibits the production of a product, and it just remains to make a form a complex enzyme, inhibitor, and substrate. There is no more production of the final product. Similarly, we can write down the mathematical equation using the law of mass action. And at quasi-steady state, where all the terms of the rate of change with respect to time become 0, then we can solve the algebraic equations. And if we rearrange the final rate of change of the product, then we have this equation. Again, this is very similar to the Meares-Menten equation. Here, the only difference is the maximum rate of change, Vmax. Vmax has been effectively changed, this Vmax prime. So you can imagine that in this case, the curves have different level of a maximum point, but with the same speed here, because Km is all the same. Not only a single binding of inhibitor and enzyme and substrate. The enzyme can also bind more than one substrate molecule, and the binding of such substrate molecules can affect the binding of another subsequent molecules. This is called cooperativity effect. We can further classify this cooperativity, either positive cooperation or negative cooperation. Here, the positive cooperativity means that when the binding of a substrate increases, the affinity of the same or different substrate. So, without cooperation, normally the curve has this shape. But if there is some cooperativity, then this profile changes to this sigmoidal curve. We can describe this cooperativity as follows. Substrate and enzyme binds together and form a first complex C1, and it can produce product. But once there is a first formation of a complex, then it makes much easier for the another formation, another binding of substrate and enzyme. So it facilitates the substrate enzyme reactions. So we can describe all these reactions using the same mass action law, and we can have this one. In quasi-steady state, the rate of change of both complex will become 0. And if we rearrange the equations with respect to the rate of change of final product, then we have this equation. In this case, if we assume these parameter values, then you will see that this rate of change of the product will become simply 2 times of the normal reactions. We can generalize this to n multiple steps, and we can further approximate. The first reaction is much slower, while the last reaction is much faster, while their product is constant. So using this assumption, then we can have the rate of change of product will become like this. If we generalize this to n times multiple steps, then we have this equation, which is called Hill equation. Hill is the name of scientist who first developed this mathematical model. And this small n actually represents the number of multiple steps of cooperativity and is called Hill coefficient. And this Hill coefficient determines the sigmoidal response curve of the resulting equation. So as n increases, as you see, the curve becomes more and more sharp. So this Hill equation is widely used to describe many molecular biochemical reactions, and particularly useful when some detailed intermediate steps are unknown, but cooperative reactions is suspected. Okay, in this lab, we are going to study mitogen-activated protein kinase cascades, shortly called MAC kinase cascades. This signal transduction pathway is very widely involved in eukaryotic signal transduction, and these pathways are conserved in cells from single-cell organism of yeast to higher organism of mammals. And MAC kinase cascades can transport extracellular stimulation such as mitogens, osmotic stress, or heat shock, and they regulate various cellular activities such as gene expression, mitosis, differentiation, and it can be very crucial in determining cell survival or apoptosis. So this is the MAC kinase signaling pathway that we are going to study in this lab, and using the previous mathematical modeling method, you can very easily develop the mathematical model of this MAC kinase pathway. And as you see here, there is a simple 3-tier cascade of signaling, and each step it has a single or double phosphorylation, and it has a feedback loop from the output to the input. You will see that this feedback actually results in many interesting dynamical properties. It's not so intuitive. So you will construct a computational model of this MAC kinase signaling pathway in your main experiment. There are a number of wonderful studies related to the signal transduction pathway. I'd like to introduce only several of them. One of them is about this NF-kappa B signaling pathway. Look at this circuit diagram. If you look this very carefully, then you'll be very intrigued, because for this NF-kappa B signaling, once it is activated by some extracellular stimulation such as TNF-alpha, then the signaling goes through all these molecular signaling cascades, and finally the NF-kappa B goes into the nucleus and it binds to its target genes. Two of them is I-kappa B alpha and I-kappa B epsilon. They are very similar isoforms, and then they produce I-kappa Bs, and this I-kappa B again binds together NF-kappa B, so it inhibits its translocation into the nucleus. So it forms a negative feedback. You might wonder why there is two apparently redundant negative feedback by I-kappa B alpha and I-kappa B epsilon. For a long time, people didn't understand why such two very similar negative feedbacks are here. But in a recent study, there was an interesting discovery that actually this looks very similar, but their role is quite different. I-kappa B alpha mediated negative feedback actually controls very different cellular response pattern. So it is a pattern designer. On the other hand, I-kappa B epsilon mediated negative feedback increases the noise level. So it can be considered as a noise maker to result in heterogeneous cellular responses. Another example about it's also related to McKinney's cascade pathway, but people wondered, because there are so many feedback regulations in this ERK pathway, and people believed such complicated feedback regulation is related to metastasis of cancer. Many cancer patients die because of this metastasis, and there was a recent study about the role of feedback regulations in controlling metastasis. That is based on computational modeling of this ERK signaling pathway. Another example is about the cell cycle control. So in mammalian cell cycle, people found that there is a bistable switch at the restriction point. And also another very interesting study is the role of feedback loops that are interlinked together and can finely tune the oscillation of molecular concentrations. So it was discovered that the coupled positive and negative feedback loops can actually produce a robust and also very tunable biological oscillations in this study. Okay, that's it for today, and I hope you enjoy your experiments. Thank you.